All right, so I am going to podcast the Chapter 15 presentation because of the ice day. And so we've talked a little bit in previous chapters about some of the different kingdoms. There are 6: animal, plant, fungus, protist. And then also there are 2 prokaryotic kingdoms, bacteria and archaebacteria. And so the focus of Chapter 15, or at least this half of Chapter 15, is going to be on the 2 prokaryotic kingdoms, bacteria and archaebacteria. Now, one of the interesting things about bacteria is how they are able to adapt to survive in so many different environments. So prokaryotes can be found on or in plants and animals, the soil, the ocean, very extreme environments, living, dead. That is not true of eukaryotes. Eukaryote survival is really limited to only a few environments. And so here is the first practice question. If you go to the Nearpod presentation, which will be in extended learning activities, you'll find these same questions. There are 10 questions, and they will be a half of a point each for bonus points. So again, here is the phylogenetic tree, and you can see that they evolved from a single-celled ancestor into 2 different domains. There are 2 prokaryotic domains and then the eukaryotic domains. And so the 2 prokaryotic domains are the eubacteria, which are the bacteria that you are probably most familiar with. They're E. coli, strep, staph, the ones that you think of that typically cause infection. The archaebacteria, however, are the most closely related to eukaryotes. These are the bacteria that live in really extreme conditions. So the thermophiles live in really extreme temperatures, either really, really hot, like in a boiling spring, or really, really cold. The halophiles live in very salty environments, which would rupture most cells, but they have evolved to be able to withstand that concentration of salt. The methanogens are found in the digestive tract of animals. So here is the second practice question found in the Nearpod presentation. All right, so now we're going to move on to talk about prokaryotic structure. Prokaryotes are unicellular, and the cells are found in 3 distinct shapes, either rod-shaped, which are called bacilli, circular-shaped, which is called cocci, or a spiral-shaped cell called spirilla. And these include spirochetes, which is the bacteria that causes Lyme disease. Now, prokaryotes are always unicellular. I probably should have labeled this differently. So they're never a multicellular organism, but sometimes they do clump together. And so a clump of bacteria that are arranged in a chain are called strepto. So if it's a chain of rod-shaped bacteria, it would be streptobacilli. A chain of circular bacteria would be streptococci. And then if they're arranged in more of a cluster, then it gets the prefix staphylo. So a cluster of rods would be staphylobacilli, and a cluster of circular cells would be staphylococcus. Prokaryotes are also quite a bit smaller than eukaryotic cells. They're about 1/10 of the size, and they're also a lot less complex. So if you look at the internal structure of a prokaryote, we've talked about this before, there are no membrane-bound organelles besides ribosomes. They have to have ribosomes to make proteins. But the DNA is not housed in a nucleus. It's actually just floating in the central region. It's called the nucleoid region, but it does not have a membrane around it. Just kind of an interesting fact. Of course, prokaryotes do not have organelles, so they don't have mitochondria. But it is thought that the mitochondrial organelle was evolved in eukaryotes by being engulfed from an aerobic bacteria that didn't digest it. And so this bacterial cell just continued inside of this new eukaryotic cell environment doing its job, which is using oxygen to make a lot of energy. And so one of the reasons why they think this is because if you look at the structure on the left of the cyanobacterium, which is a photosynthetic bacteria, it very much resembles the chloroplast. It has a highly folded membrane. And so the thought was perhaps a eukaryotic cell engulfed this process called endosymbiosis, engulfed this photosynthetic bacteria so that now these eukaryotic cells had a way to use light as a source of energy. And then mitochondria would be a eukaryotic cell that engulfed an aerobic bacteria. So, and I think we talked about this in one of the articles I gave you last semester, that also mitochondria and chloroplasts do have DNA, and so that further supports this theory that they were once their own cell that have been engulfed and then just persist in being passed down every time a cell divides. Now this new organelle is just being passed down through the generations. All right, so just a few other structures. So if you look at this picture of a prokaryotic cell, you can see the DNA in the nucleoid region. You can see ribosomes, no other membrane-bound organelles. We talked about the cell membrane and also the cell wall that surrounds prokaryotic cells in a previous chapter. But also, prokaryotes sometimes, not always, in addition to the cell membrane and cell wall, are also enclosed by a capsule, which just provides additional protection. Another source of protection is the endospore, which you can see in this picture at the top. It's a thick-coated protective cell that's produced inside of the bacteria, protecting the DNA, so the genetic material, from being decomposed in any way, so that when this bacterial cell is exposed to harsh conditions, it can still survive. It's dormant, and it can be dormant for years and years, but the cell itself will survive, and when the conditions are not so harsh, that cell can then begin to replicate the DNA and make the proteins that are necessary for survival. Now, these are not the archaebacteria. This is a mechanism that evolved in the eubacteria in the event that they did or were exposed to really harsh conditions. And then the last thing I wanted to point out were these projections from the cell wall called pili. Again, this is not found on all prokaryotic cells, but this is just a structure, just these short projections that can help prokaryotic cells stick to surfaces. Looking at the structure of the prokaryotic cell, they can be classified as gram-positive or gram-negative. And so that classification is based on the amount of peptidoglycan that's in the cell wall. And so it's just a technique that scientists and medical professionals use to identify bacteria. They gram stain it, and then they classify it as gram-positive or gram-negative. So, for example, strep is a gram-positive bacteria. So when that culture is taken from your throat, if it's gram-positive, they've narrowed it down to a few options, one of which could be strep. And then also, bacteria, of course, reproduce asexually about every 20 minutes through a process called binary fission. So it's just a process where prokaryotes copy themselves and then split in half. And the DNA, just like we've talked about in previous chapters, has to be replicated for the new cell. And the mutations in the DNA account for the genetic variation in bacteria. So there are quite a few questions, I think 5 total questions at this point in the Nearpod. And then we're going to move on to the section where we talk about prokaryotic metabolism. So how do bacteria acquire their energy? And so there are 2 broad categories. They're either autotrophs or heterotrophs. Autotrophs mean that they acquire energy on their own. And so they can either do that photosynthetically, that would be a photoautotroph, or a chemoautotroph uses inorganic compounds such as carbon dioxide in the absence of light, and they use those to build organic molecules necessary for life. So autotrophs acquire energy on their own. Heterotrophs have to acquire energy from another living source. So they live on plant and animal matter. It could be, it doesn't have to be living, it can be a dead plant or animal, in which case the bacteria is a decomposer, but it requires another living source of energy. So here's a practice question. And then finally, we finish up this chapter talking about the ecological impact of prokaryotes. There are, of course, a number of bacteria that cause infections. Those are called pathogens. And most of the bacteria cause disease by producing poisons. So the poisons can either be secreted through exotoxins. These are just poisonous proteins that are secreted out of the bacterial cell that make you sick. Or an endotoxin is a chemical component of the bacteria itself, typically in the cell wall, that makes you sick. And so food poisoning is an example of an illness that is caused by bacterial endotoxins. Bacteria don't have to be harmful. They're not all pathogenic. In fact, life on Earth is dependent on bacteria. They're necessary to recycle nutrients. They're necessary to fix nitrogen from the atmosphere. So nitrogen, when we talked about proteins and when we talked about DNA, both of those have nitrogen in them. And so nitrogen is necessary to synthesize proteins and nucleic acids, both of which are essential to life. And all of that nitrogen comes from the nitrogen fixation of bacteria on the nodules of plant roots. So without bacteria, we would not have a usable form of nitrogen, and we would not exist. Also, bacteria can be used to remove pollutants from water, air, or soil. Using bacteria to clean up the environment is called bioremediation. And then finally, bacteria is used to produce a number of foods. Of course, yogurt is a great source of bacteria. But bacteria are also used as factories to produce medicine. So insulin is a drug that is produced in E. coli bacteria, which has brought the cost down considerably because now it doesn't have to be harvested from live animals, which in the past was how it was obtained. Here are a couple more practice questions. And then finally, just to close, Craig Venter is someone that we talked about when we talked about the Human Genome Project, but his latest venture is that he created a synthetic form of life that is going to be a source of alternative fuels. and